Hello, everyone, and welcome to Neurocracy. Today, we're joined by Dr. David M. Raizen, a professor of neurology at the University of Pennsylvania who studies the regulation of sleep using the model organism C. elegans. His research focuses on sleep-like states during development and stress, with the goal of understanding why we sleep and how it evolved and how it's controlled. Dr. Raizen, thank you so much for joining me today.
Thank you for inviting me. This is a real honor. Thank you.
Yeah. So, what first drew you to studying sleep?
I probably have been like the rest of us. I've been interested in sleep from as long as I can remember.
Yeah.
The things that caught my eye in particular when it comes to the clinical side of sleep is when I was a neurology resident, I would do consultations in the intensive care unit on patients who had altered levels of consciousness. Sometimes they were in coma.
Yeah.
And I started to read a bit about the circuits that mediate arousal and stupor and coma. And I realized that that literature overlapped tremendously with the literature about the arousal systems during sleep and wake cycles. So my first clinical attraction to this, and so as sort of as a clinical subspecialty, was as a neurology resident. And of course, I've gotten back to the lab and back to the very basic science when we're studying sleep. But the original motivation was probably clinical more so than anything else.
Okay, that's interesting. So you do a lot of research with C. elegans. Could you talk a little bit about how you kind of started doing that and why C. elegans is such a powerful model when we're studying sleep?
Yeah, so when you want to study a biological process, you want to try to study in the simplest possible system that you can. And a great example of the power of using a model system to understand basic biology comes from work in the 80s and 90s on cell cycle, primarily in yeast, single cell organisms. And that understanding of cell cycle and growth regulation and signaling done at this very minimalistic model system turned out to be very relevant to our understanding of cancer and when cells don't divide properly.
Yeah.
So it demonstrates the power of using a very simple model system. And when it comes to sleep, the simplest model system that's in use, I would argue even today, is this tiny nematode Caenorhabditis elegans. The advantage of C. elegans is that it's small, it's very easy to cultivate in the lab. It's got a very simple nervous system, consists of just 302 neurons, and we know the names of, since it's such a small number of neurons, we actually have a name for every single neuron. We have a name for that. And you'll probably hear more about that in my interview today. But
Yeah.
that is a real minimalistic sort of system that I thought would be very powerful in pursuing mechanistic questions that have to do with cell signaling and with circuit analysis and things like that. So Drosophila, when I started working on worms, Drosophila was already established as a model for sleep research. But Drosophila, at least at that point, had a very powerful um, assay for monitoring sleep by putting a fly in a little tube. And the genetics were very powerful in the fly, but at least at that time, the neurocircuitry in flies was not understood, nowhere as close
Oh,
as it
yeah.
was C. elegans. So C. elegans offered that extra advantage of having a very literally transparent nervous system because the animal is
Yeah.
actually non-pigmented, so light can get in and light can get out. So you can monitor fluorescence of genetically encoded fluorescence sensors. You can monitor any cell in the body very easily just with a light microscope.
Yeah. So what neurons, out of those 302 neurons, which ones stood out to you in terms of their function and ability to regulate sleep? And I was looking at some of your work and I noticed some interesting,
Yeah.
yeah.
So this is actually work by my colleagues in the field, but the two neurons that have sort of stood out in terms of their importance to sleep are one is called the RIS neuron,
Yeah.
and the other one is called the ALA neurons. They're both single neurons. They're not, some neurons in worms are paired, so they're not paired, they're just single neurons. And they are second order interneurons, meaning that they don't receive direct information from any sensory neurons, but they respond to two synapses down. So like the sensory neuron talks to a first order interneuron, and that first order interneuron talks to a second order interneuron. And RIS and ALA are second order interneurons. So the reason I'm emphasizing that point is because you can think of them as the closest thing to be deep brain structures in humans, right?
Okay. Yeah.
We think of like the hypothalamus, which is a part of our brain that controls a lot of our homeostatic behavior such as sleep, we can think of that structure as being sort of s- deep in the brain, central
Yeah.
nervous system.
Yeah. So, what has some of your like projects or work been regarding these neurons?
Well, we started our research in worms studying sleep during development, and the attraction for development is that there the there's a famous circadian protein discovered in Drosophila called period,
Oh
and
yeah.
period turns out to regulate circadian rhythms not just in fruit flies but also in humans. But in C. elegans, there is a period, what we call a period ortholog, a protein that looks like fruit fly and mammalian period. But it doesn't oscillate its expression in circadian or 24-hour rhythm. It oscillates its expression in a molting rhythm. So when the animal molts, this gene,
Oh,
like protein, goes down. When it's in the active phase between the molts, it goes up. So it oscillates and it maintains that same relationship that's shown between Drosophila period and Drosophila sleep as between C. elegans period and C. elegans quiescence. We didn't call it sleep back then, but we call it quiescence, lack
yeah.
of movement.
Yeah.
So the worm develops from an egg to an adult through four larval stages, L1, L2, L3, and L4. And during that transition between larval phases, that's when the worm, we think, sleeps. That's when it becomes quiescent. It's less responsive to the environment. And that sleep is homeostatically regulated in that if you prevent the animal from getting it, the animal will then sleep longer or deeper when you eventually allow it to sleep. So back then, that was the primary interest of ours, is to study what we now call developmentally timed sleep, and we think it's very
Yeah.
similar to circadianly timed sleep in humans. The main neuron involved in that developmentally timed sleep is the RIS neuron, and that was demonstrated by a colleague in Germany, Henrik Brigman. And but then when we pursued our experiments, we came upon a sort of an odd observation that took us in a different direction. And that observation was that if we stress the animals, if we for a long time we thought that they didn't sleep during adulthood, they only slept during this developmentally time period during larval development. But we found that if we took adults and stressed them, by stress I don't mean like psychological stress, but cellular stress. Like
Okay.
if we expose them to high temperatures or to ultraviolet light, which is thought to damage their DNA, they will initially try to escape, but then they will actually stop moving and go to sleep. Okay.
Wow.
And we called that cell stress induced sleep or sickness induced sleep or sleep in sickness. The reason we chose the name SIS that was pretty generic is because you can fit various words to it.
Yeah.
But um, but that sickness sleep has gotten sort of our primary attention now in the lab. We study that closely, and the neuron that we think RIS neuron is involved in sickness sleep, but there's another neuron, ALA, that its only role that we can identify is involvement in this stress-induced sleep or SIS. We've pursued this not only because it's interesting, which of course it is.
Yeah.
Much of what we do based on curiosity
Yeah.
and trying to understand nature, but also because we think it's very clinically relevant to um, to the kind of sleep or the kind of fatigue that you get when you're sick,
Yeah. Yeah.
right? We've all had COVID or flu or at some point
Hmm.
in our life,
Yeah.
and there can be different manifestations to the illness, whether it's a respiratory illness, you get coughing and shortness of breath, or if it's a intestinal illness, you can get a gastroenteritis.
Yeah.
But what's common to all these diseases is that you get profoundly fatigued, and right, so if
Yeah.
you recover from the flu, you don't want to go out, hang out with friends. You don't want to eat meals. You just want to go to bed and rest. And so that's a normal adaptive behavior to sickness, which all animals do, including us and including C. elegans, as it turns out. But it's clinically important, not so much because we want to learn how to get rid of the fatigue that's associated with acute infection, but because there are people who continue to be fatigued even after the infection has gone on and has resolved. And those patients are incredibly disabled by their fatigue. and we really don't have a mechanistic inroad to understand what causes their fatigue. So this is where we think that this model in C. elegans studying the sickness-induced sleep will be relevant to our understanding of fatigue in people.
Yeah, I think that's very interesting. I mean, I know whenever I have a cold, I always just can't seem to get out of bed, and I seem to be sleeping a lot more too. Funny enough, actually, I remember I sprained my ankle back in November, and I had this feeling that I was getting just a few more hours of sleep every day, just a little bit. And I have like a feeling it was somehow related to my ankle. And I remember I was actually talking to someone on the podcast about this at the time. But I think it's very interesting to see the clinical like applications of that sickness-induced sleep.
Yeah.
So, actually, I had a question about your research with the developmental stages and the effects or like correlation with changes in sleep. So I know one thing that I personally like advocate for is pushing school start times back because teenagers have that biological clock shift of 2 hours. So I'm curious, is there any correlation between life stages in humans corresponding to sleep shifts and what we observe in C. elegans in terms of those developmental changes?
Yeah, no, that's a really interesting thought. I actually have never thought about that the worm research in those lines.
Yeah.
Uh, the the worm doesn't have circadian rhythms, at least not robust ones that we can detect, right?
Okay.
So the humans and fruit flies, a lot of organisms have very prominent circadian rhythms. That's why we sleep at night and we're awake during the day.
Yeah.
Uh, and it's the phase of the circadian clock which promote wakefulness that changes during our teenage years, and that's why, as you point out, we tend to like to go to sleep later and
Yeah.
wake up later during our teens. Um, so worms don't really have. I mean, they have a developmental clock, and that controls the
Yeah.
timing of their larval molts and the timing of their developmentally timed sleep. But I don't know if there's any so I guess the analogy to the human advance or delaying of the sleep phase would be if somehow the sleep got longer or extended, its onset started later and during I don't know. I have to give
Okay.
more. It's a
Yeah.
real question.
Yeah, just something that came to my head because it's always in the back of my mind, like teenagers, because I'm a teenager, so that's one of the things that I'm always thinking about. So this stress-induced sleep, like like it um it likely developed through evolutionary like pressure, right? So I'm just curious, like what are some explanations for what might be going on during this stress-induced sleep that might be like different than, just like the obvious like resting. Like I'm just curious, like are there any like interesting mechanisms that might be at play here?
No, I think you're asking beautiful questions, and I think that's really at the crux of this behavior that we're studying is why did the animal develop to engage in this?
Yeah.
We do know, and this is an experiment I think you can uniquely only do in the worm, which
Okay.
is test the effect of curtailment of that stress-induced sleep. What's the consequence of getting rid of it to the animal's physiology? And specifically in worms, because they only live 2 to 3 weeks, you can
Yeah.
actually use mortality as the endpoint of study.
Oh yeah, that's true.
And if you do that, my colleague in California, Dr. Van Buskirk, did that experiment with heat by heating up the animals, and then she got rid of sleep by using a mutation. And what she found is the animals that were heated up and could not sleep were much more likely to die than animals that could sleep. So there really clearly is an advantage to sleeping that is it's not just a correlate, but it's actually playing a role in the recovery from illness. We've shown the same effect with a viral infection model. So if we infect the animal with a virus and we prevent the animal from sleeping, they also they die earlier. So we think that's evolutionary adaptive. Now what the benefit might be, we don't yet know.
Okay.
One thought that we had and we still have, and others have pursued this idea also, is that this sleep is involved somehow to help you energetically to deal with the insult. So, for example, heat shock causes proteins to malfold and form aggregates, and if you getting rid of those aggregates is very energetically demanding, requires a lot of ATP.
Yeah.
So maybe sleep is facilitating that recovery, and that's the same thing with a viral infection. The part that doesn't quite fit right for that, though, is that when our animals are asleep, and it's not only that they're not moving and they're less responsive to the environment, but they're also not eating. They stop eating.
Oh, okay. That's
So
interesting.
they may be gaining something from sleeping or being inactive, but they're losing something by not bringing in as many calories when they're sleeping. So
Yeah.
it's there's probably a happy medium, like sort of um, a certain amount of activity is probably good because it allows them to eat, and a certain amount is bad because they don't eat. And it's somehow there's some sort of energetic equation there that we think goes awry when the animals are injured, and they must get that rest in order to recover. But what the energetic advantages, we still don't know. We're really interested in that question, as are others.
Is there possibly an increased like hunger drive during wakefulness to maybe compensate for that, or has that not been like studied yet?
Has not been studied. I think
Okay,
that's a
yeah,
great question. There is, acutely when the animals or get exposed to the ultraviolet light, they actually don't go to sleep. They actually try to escape, and maybe that's when they eat a lot. We don't know. We don't know if they eat excessively when they come out of their sleep state because they
yeah.
sleep only transiently. The heat shock will cause them to sleep about an hour. The UV exposure maybe 4 hours, but they do come out of it and. That's a really interesting question. Maybe they eat voraciously when they came out of it, sort of like probably after you sprained your ankle, you probably were
I
hungry and
yeah, I did have interesting food habits actually. Yeah, I remember I was trying to eat as much protein as I possibly could because
okay.
I was under the impression that as much protein as I could get would be help with my recovery. And I'm not sure what I did right, but I mean, it's the recovery has been going pretty well. So I think whatever I did worked. But yeah, so I actually had a question about these stresses that we are talking about. Like for example, like heat shock for C. elegans. Are there any like parallels for humans? And I know when we're talking about stress, we're not talking about psychological stress here. I'm just curious, like, for example, like one thing I'm thinking of is like radiation exposure might be
Yeah.
something. Like I'm just curious what kind of things do we
Yeah,
see
I
for
think I think
humans?
that's probably the first thing that pops to my mind as well as radiation exposure, because what
Yeah.
radiation does, ionizing radiation does, is it causes damage to the DNA.
Yeah.
Okay, and
Yeah.
ultraviolet radiation also causes typically double-stranded breaks to the DNA, and that needs to be repaired. Um, so um, So in terms of the human type exposures that can trigger the same sickness-induced sleep, I think ionizing radiation that's used to treat
Yeah.
cancer, for example. In fact, one of
Oh,
the
yeah. That's actually
most disabling side effects that cancer patients who undergo radiation therapy, the side effect that bothers them the most, they talk about is fatigue. Um,
Oh, I
they
actually
talk
didn't know that.
about
Okay.
is fatigue. In fact, that was one of um, the population of patients I see in the clinic who come to me they're referred to me by oncologists because they're profoundly fatigued during their treatment, not just with radiation, but also with chemotherapy. That's also genotoxic. That's also injurious to the DNA.
Yeah.
So that would be one example. Another example might be patients who have rheumatological disorders. I don't know if you've heard of rheumatoid arthritis or lupus
Yeah.
erythematosus. So these are conditions that we think of the inflammatory. The immune system is hyperactivated. There's an autoimmune action. And there's release of cytokines, which are small proteins in the blood that get released and travel in the circulation. And those patients are also profoundly fatigued. Again, they're not necessarily sleepy, but they're fatigued. They
Yeah.
really have making it through the day. Um, so, so I think those are probably the most obvious analogs. Oh, the third one, which you actually brought up with your sprained ankle anecdote, is that tissue injury of any kind causes fatigue and sleepiness. So
Oh, okay. So I wasn't making it up. There's definitely
I
some.
don't know. People
Yeah.
who have gone through surgery,
Okay.
uh, when they wake up out of surgery, of course, that's complicated because they're sick with something. They
Right.
get, anesthetics. There's a lot of variables that have
Yeah.
changed. But invariably, people who come out of surgery are profoundly fatigued for a few days. So the trauma to the body that an incision of the knife does, that can cause fatigue. And in fact, that's been modeled in C. elegans,
Oh,
where if
wow.
you
Okay.
take a little needle and give a little scratch on the cuticle, so you break the cuticle of the animal, that animal then goes to sleep for about an hour or two
Hmm.
until it can repair things and wake up. So So yeah, so there's a lot of analogies between these, anything that causes cellular stress, be it heat exposure or ionizing radiation or trauma or inflammation. Those are all causes of stress-induced sleep or sickness-induced sleep.
Yeah. So I'm curious more with like the like cancer patients with the increased fatigue after radiation or chemotherapy. So does this like fatigue linger after like treatment might like maybe after like their treatment ends or like is are there like interventions for them to help manage their fatigue? I'm just curious like how that but
Yeah.
also at the same time like I feel like that fatigue might be serving like some purpose. So is there an argument to be made that there shouldn't be an intervention too? So
Sanjay, you're asking all the right questions. You're ready
yeah.
to skip right to being a doctor. You don't need to go to college or medical. No, those are great questions. So, uh, some patients who get chemotherapy, who get radiation therapy, for most patients, once they're done with the therapy, they feel better, they recover. But there are some patients who continue to have fatigue, and they go through sort of a transformation, which is initially more like sleepiness, sort of acute fatigue, to more of a chronic fatigue, and feeling like they're sick all the time, even when they're sick, they're not sick.
Yeah.
And that's a real syndrome. That's a disease called chronic fatigue syndrome. It's got even a longer name than that, but that's
Yeah.
kind of a popular way of referring to it. And it's very disabling, and it's surprisingly common. There's estimates the government put together a task force sponsored by the Institute of Medicine a few years ago, about a decade ago, maybe a little less, and they estimate that it depends which study you read it, maybe 500,000 to even 1 million Americans have some degree of chronic fatigue syndrome, which is a
Wow.
staggering number of people. And
Yeah.
I think they're probably we probably don't even know a lot of people who have this because they're so fatigued they can't even make it to the doctor's office, so they just they
Wow.
don't seek medical attention.
Yeah.
But let's come back to your other comment that you made there, which is that should we be treating their fatigue, and
Yeah.
is there a way to treat it? Um, Unfortunately, right now we don't have good ways to treat the fatigue. If there's a sleep disorder associated with fatigue, then we try to treat the sleep disorder. Like if
Yeah.
there's the airway is obstructive, if there's sleep apnea, if there is insomnia, if there's narcolepsy or something like that, then we try
Yeah.
to treat. But for your garden variety fatigue, you're not going to have a treatment. But then gets back to your question, should we be treating it? And maybe it's providing a beneficial effect to the animal being us, humans. But so we don't know. I think if we come up with a treatment, I think we have to be very careful about how we administer it and to whom do we administer it. And we have to think hard about, will this treatment actually cause harm? Because the body's natural reaction to the physiological stress is to stay immobile and move less and be fatigued. So
Yeah.
yeah, so we don't know what's going to happen if you treat it. Well, my hunch is that acute fatigue you never want to treat, but
Okay. Yeah.
chronic fatigue, because of it's all cost benefit, because the cost is so high of having chronic fatigue.
Yeah.
Maybe it's worth looking at that in terms of a disease that can be treated. At least the fatigue aspect can be treated and prevent the
Yeah.
chronic phase.
Yeah. So, as a high schooler, I'm surrounded by friends who like, and I don't really do this too much, but a lot of my friends, like, if they're having like sleep problems, if they're feeling really tired, they'll turn to caffeine, like Red Bull, Celsius drinks for like an easy fix, or sometimes even melatonin. And I personally never found like any appeal for that. I just like if occasionally, like if I have to stay up a little late, I'll push through. But I do definitely know a lot of people my age like use those kind of ways like a cheat code almost. And I'm curious, like maybe if someone has this chronic fatigue syndrome and they're like using this melatonin or like caffeine intake to try to compensate, what kind of happens there? Have you seen this before? Or just
Oh yeah,
curious.
when people are fatigued, they'll try anything. And
Yeah,
obviously caffeine is one of the first things that they try because it's cheap, it's available. You don't need a prescription. It's the number one. If you think about it, it's the number one pharmaceutical in the world, caffeine,
yeah.
right?
Dr. Alan Pack said that in his.
Yeah.
I just interviewed him just a few weeks ago. He said that quote unquote. He said Starbucks is the biggest pharmaceutical in the world. So, yeah.
Dr. Pack was my postdoctoral advisor, so maybe
Oh
some of his
wow.
maybe
Oh,
some
that's
of
funny.
his
That's
rubbed off on
interesting.
me.
Yeah.
So.
Yeah.
Yeah, so Yeah, I don't think they see much of an effect of caffeine. Sometimes caffeine will help people stay awake, but doesn't actually give them energy, and it's not a productive wakefulness. It's not,
Right.
they're still in a brain fog. Sometimes we call it brain fog or trouble thinking, but the caffeine may just prevent you from being able, like your friends who take it, if they were to not take it and just lie down and took a nap for an hour or 2 hours, they'd probably be much better off than, because the caffeine is not going to substitute for their sleep. It's going to
Right.
maybe
Yeah.
prevent it for a little bit, but then they're going to crash.
Yeah. And also I think the other thing with caffeine is that while it may like curb that adenosine, like that homeostatic drive, I think it actually rebounds more because it doesn't take away that underlying fatigue like after that initial like wave wears off. Do you have any like thoughts on the use of melatonin, for instance, in like trying to curb this chronic fatigue and how does that like tend to work?
Yeah, so melatonin is interesting. It's found pretty much throughout phylogeny, even I believe even in jellyfish, Cnidaria,
Oh wow.
and C. elegans. I don't know if it has melatonin, but it does have melatonin receptors.
Okay.
They're involved in regulating sleep in a very loose way. So it's a very ancient hormone that seems to function. I think I have this recollection. I've actually seen some literature that's also expressed in plants, but I may be wrong about that.
Okay.
But what's interesting about melatonin is in some model organisms like songbirds or fish
Yeah.
or flatworms, the melatonin will put those animals to sleep very acutely. Like you give them you add melatonin to the bath and they'll of the fish and they'll just stop moving.
Wow.
But in humans, the role of melatonin is a little bit more nuanced, and it's more of a what I think of as a chronotherapeutic. It's in training our circadian rhythms rather than actually acting as a sedative hypnotic, putting
Yeah.
us to sleep directly. And there's been a lot of literature on this, a lot of reviews of the literature. I think that's probably the best consensus the field has right now, that melatonin is more of a I don't know if in your interviews you came across the whole story of process C and process S. Does that ring a bell to you?
Yes, that does. I never t talked about it in an interview, but I have like heard those like concepts before.
Process C is
Yeah.
the circadian process that
Yeah,
regulates
yeah,
alertness during the day. And we think of melatonin being involved in process C,
yeah,
not so much processes, not so much sleep drive. And but so a lot of people take it to help them sleep or try to help them sleep by taking it. Let's say they stay up till 2 in the morning, they'll take a melatonin and try to help them fall asleep. The problem with taking melatonin at 2 in the morning is that your endogenous levels of melatonin normally go up at night and are probably at their peak at 2 in the morning. So taking a pill that has melatonin in it is really not going to affect
yeah,
your melatonin status because you already have melatonin in your system. What it might do if you take it that late is actually delay your sleep phase in the morning because the pill that you take may linger, the effects may linger in the bloodstream for maybe 4 hours or 5 hours. So normally you might want to wake up at 6 in the morning, but then the melatonin you took at 2 in the morning keeps you asleep for longer. So that
yeah,
can be so yeah, so I think the best use of melatonin, at least clinically, is more as a time giver, as something to tell our bodies when it's time to go to sleep, and
yeah,
usually take it much earlier than the nighttime. So if not, if this time of year in Philadelphia it gets dark around 5:30 p.m. But if you go to sleep at 10 p.m. or 11 p.m., you want to take the melatonin earlier. You don't want to take it right when you go to sleep because the lights are already out, your melatonin levels are already up. So if you're trying to advance your sleep phase, let's say if you're a teenager and that clock is being delayed and you're trying to advance it in order to get to school on time, that's when you would take it a little earlier when it's still light outside.
interesting. Yeah. I think people always have like differing opinions. Like my friends, as in like people who aren't like sleep experts, like always have like different stories on how melatonin helps and not. And I've actually heard that the placebo effect is also like very powerful when it comes to melatonin as well.
Yeah,
So, yeah.
I'm not opposed to the placebo effect if it
Yeah.
works. I don't care if it's placebo or
Yeah.
not placebo, but in terms of doing a study, a scientific study, it's you have to control for that placebo effect. Of course, you're right.
Yeah, yeah. So, looking forward in the next couple of years, what kind of experiments are you excited to continue doing with C. elegans, in terms of sleep?
Yeah, so I don't think I emphasized this in the beginning, but I think it's important to remember that the worm is what it's really, really good for. I mean, it's great because it's got a simple nervous system. It's got a fast life cycle. It's got this sleep, developmentally timed sleep and stress-induced sleep. But what it's really a remarkable organism for doing is what we call discovery science. Not hypothesis-driven science, but discovery science. So hypothesis-driven science would be something like, I'm going to take this gene out and ask, what does it do to the circadian rhythm, or what does it do to the developmentally timed sleep? So
Yeah.
an example of that is that gene that I told you about being similar to period.
Yeah.
But discovery science is one where you're coming in not with a hypothesis as specific, but you're trying to gather information that will be hypothesis generating, which will
Hmm.
lead you to think of certain
Yeah.
hypotheses. And the information, the way we do these experiments, the most common way we do them is we do what we call forward genetic screens. Okay, so we mutate genes randomly in the genome, and then we ask, what is the consequence? So then we get these colonies of mutants. Okay, so and then we ask, do these mutants have a sleep phenotype? Are they now excessively sleepy? Are they now unable to sleep? Once we find a mutant that's unable to sleep, for example, then we triangulate. We do what's called genetic mapping and
Yeah.
sequencing to identify the gene that's mutated to cause that phenotype. And then once we find the gene, and we've done this a number of times, let's say it looks like the gene looks like a receptor, a cell receptor, which is one of the genes we found. Then we study it in the circuit. We try to figure out where in the animal it acts, when it acts, and then of course we're doing this discovery genetics also to find new things about us humans. So if we
Yeah.
find a result in C. elegans, the next step may be and it's robust results. So the next step may be to try it also in fruit flies or in mice. Okay, so those will be other translational model systems, and eventually try to bring that to humans, either with genetics, human genetics, or some sort of bioassay for the chemical. But of course, our ultimate goal is to help um, humankind, and that requires some sort of translation from the worm up to humans. So in terms of what we're working on, we're doing genetic screens. We're actively doing that right now. We have publication of several genes that we identified that work together that we're trying to write right now.
Yeah.
There's another gene that one of my grad students is working on. So that's one approach for doing discovery science and C. elegans. The other approach is the ultimate idea is to come up with knowledge that will then lead to a therapeutic in human, right?
Right. Yeah.
And that therapeutic is typically a small molecule, small synthetic molecule that pharmaceutical companies build. Okay, maybe a molecule that looks like caffeine if we're trying to keep people awake, or a molecule that looks like melatonin if we're trying to give us chronotherapeutic. But you can also skip that step and you can go straight to the chemicals. The C. elegans system allows you to screen large libraries of chemicals very rapidly.
Oh, yeah.
And you can take a library. We actually recently just did this. We took a library of around 4,000 chemicals
Wow.
and we tried each one of those chemicals on worms and asked, does any of the chemicals block the ability of the animal to fall asleep during this stress-induced sleep? And we found quite a few that did. In fact, we found 42 out of the 4,000, about 1% or 42 had this disruptive effect on sleep. So then we study that the C. elegans system allows us to study what's the mechanism that these drugs are affecting sleep. What are the cells that they're going through? What are the molecules that they're going through?
Yeah.
So that's another form of discovery science that we do in C. elegans that's a unique power of this model system you can't easily do elsewhere.
Yeah, and I'm actually glad that you emphasized that discovery science. I think I remember when I used to like I mean I still do, but like I remember when I was like first studying biology, I came across like those like two branches of science where like one is more like discovery based where you're not really sure where you're going, but you're making sure to like continue to be curious and ask questions that can lead to like hypothesis driven science. And I think sometimes it's easy to like forget about that the importance of just asking questions and going and finding like the unknown so that down the road people can form hypotheses and open up new branches because I think we can both agree that some of the most like interesting and like most famous discoveries in science have been by chance or unexpected experiments so
Absolutely. And by the way, do you know how science is like Christopher, how is doing science like being Christopher Columbus? Have you heard this analogy?
No, I have some ideas about what it might be, but no.
Christopher Columbus took a boat and went west, right? And he arrived in the New World, and then when he got there, he really didn't know where he was. He didn't know where he was going when he set out. When he got there, he didn't know where he was.
Yeah.
When he got back to Portugal, he didn't know where he's been. And to top it all off, he got the federal government to pay for it all, right?
Yeah.
That's the analogy we have in
That's
the
that's funny. Yeah. Yeah.
well, nowadays, the funding levels are getting tight even for American scientists. But for a long time, the government was a wonderful funder of basic science, which is the
Yeah,
core.
actually, I wanted to add there. I remember I was speaking with a scientist. I forget who it was on the podcast, but I was talking with someone about how like policy and science, how sometimes in like right now, what's happening is that policymakers are like funding certain projects based on how cool they look or on outputs that they hope to find based on like what direction that project might appear to be going in, and they're cutting funding from like discovery-based science because it doesn't seem to like attract as much like stakeholders, for instance. And
Yeah.
I just think I remember they were talking about how it's important to like let science like progress naturally rather than picking and choosing what experiments sound good based on that hypothesis driven science as opposed and just like if you want to like add anything about that too.
No, I absolutely. And
Yeah.
the the classic study that was done by in the laboratory of Seymour Benzer back in the 70s, was published in the 70s. His student Ron Konopka did one of these discovery approaches. He looked for mutants that no longer had altered circadian rhythms, and that's how he found the gene period, which that was really esoteric science back in 1971.
Yeah.
Hardly anyone cited that paper that they published in PNAS, but then years later, that little study in Drosophila led to our understanding of um, the circadian clock at the molecular level, and we understand it so well, in fact, that the Nobel Committee in Sweden decided to award the Nobel Prize for physiology and medicine to the circadian researchers that worked this out back in, I think it was 2017 when they gave that award. But it's
Wow.
based on a very basic scientific approach, which was discovery genetics, a genetic screen done in fruit flies back in 1971.
Yeah. And I also have like a personal connection with period because I remember my internship at WashU my year after freshman year we were look I was helping someone with the project that was analyzing the role of the Nalcyon gene in maintaining circadian rhythm periodicity. And I remember we were also doing some stuff with a fusion protein with period gene with the firefly luciferase so that we could like monitor the glow and the expression of the period gene. So that was definitely like that gene is always something that will be in my mind. But yeah.
That sounds like a wonderful internship. I'm jealous. I wish I'd done that when I was your age.
Yeah. Yeah. I mean, I was very fortunate. Dr. Herzog was a very, very great mentor here at WashU. But yeah. So on that note, Dr. Raisin, thank you so much for joining me today. It was really fun talking with you. And now I know a lot more about how C. elegans are able to provide insights on sleep.
It was delightful to be here. Thank you. Bye-bye.
Yeah.